Okay, in our case study today, we are going to be talking about Oncovin. It is a cancer drug to treat cancer patients in addition to other drugs, but we're going to be focusing on this one today. We're going to talk about how this drug binds to tubulin in the cell and leads to prevention of cell division. And then we're going to talk a little bit about some background information about tubulin and how it is used in cell division. So tubulin is a protein that's found in all eukaryotic cells. Alpha beta tubulin dimers assemble into microtubules. And they have a lot of functions, but most specifically in mitosis, and that's what we're going to be focusing on today in our case study. So a lot of ligands will bind to tubulin and influence its assembly properties. Among them, several drugs that have proved to have anti-cancer properties. So overall, tubulin is a protein that we're going to be focusing mostly its function and use in mitosis. So how exactly is it used in cell division and in mitosis? What is that main role that it plays? It polymerizes into long chains of filaments, which makes microtubules. So microtubules are part of the spindle apparatus, which the spindle apparatus is, of course, those spindle fibers that are on the opposite poles of the cell when mitosis is taking place. So in cell division, chromatin moves towards the pole of the equator, and that spindle apparatus helps it to move. So tubules make up microtubules. Microtubules are that movement portion. So when the chromosomes are separating, the spindle fibers, which are made up of microtubules, which is made up of tubulin, are what's helping to move it. So it has a big role in mobility, or should I say motility, inside of the cell during mitosis. So you can see here in these photos, so we're seeing the microtubules are all made up of microtubules, and they assist with the chromatin moving to those opposite ends and helping with cell division. So how is Oncovin binding to tubulin? It is an inhibitor of microtubules, so it directly inhibits microtubules. That's its main sort of prey that it goes after when you take this drug. This leads to mitotic arrest and cell death. So that's sort of the main purpose of taking the drug. So it blocks mitotic progression, and it binds to beta tubulin subunits at the positive end of microtubules at a region called the vinca binding domain. So if we see that here in a photo, so this is sort of a lot going on in this photo, but what we're focusing on here is that vinca binding site. So that is on the microtubule, and you can see here these green ones are the beta microtubules. So the vinca binding site is this this gorgeous structure, and that is where Oncovin is binding. So when Oncovin binds to the microtubule, the simply binding to it decreases the dynamic of the microtubule movement already, right? So when we're binding here, the microtubules movement and motility, that motility ability is already hindered just by having something additional binding to it, because it's not made to do that. And when we're talking about mitosis, we're talking about cell division. This is a very complex and very specific process. So if one thing is starting to go wrong, and there's additional add-ins that you're seeing going on in that cell division process, then that's immediately a no-go, and it doesn't allow it to continue to go through the rest of the cell cycle. So there's the binding aspect, and the proper assembly of the mitotic spindle is prevented, right? So it can't assemble properly, and that tension at the kinetochores is reduced. So when we look at this photo again, that kinetochore, right, here, and in this middle where it goes in between in those chromosomes, that is hindered as well. And there's tension in there that causes them to pull apart. So without that tension, there's no pulling apart process, thus there's no cell division. So unwanted cell division thus is controlled, which benefits cancer patients, because then these cancerous cells are not continuing to grow and multiply. All right, and then here is my citation section, and that is the end of my presentation.